Hello, everyone. Thank you all so much for joining us today for the NOAA Omics Seminar Series. We're going to start promptly at noon Eastern Time, so in about 2 minutes. All right, we're going to go ahead and get started. So hello, everybody. Thank you all so much for joining today. Welcome to the NOAA Omics Seminar Series. I'm your host, Catherine Egan. I am the NOAA OAR Omics Coordinator, and I sit in NOAA Ocean Exploration. I also sit on the NOAA Omics Working Group as a subcommittee co-lead. Omics, which is a suite of tools used to analyze DNA, RNA, proteins, and metabolites, has become a large priority here at NOAA in the last several years. We established the seminar series in an effort to increase transparency and collaboration and highlight the incredible omics-related research currently underway both within and outside of NOAA. The seminars from this series are made available on YouTube and posted up on our Omics website. There's a link to the website in the chat now for folks who want to go and view past webinars. As participants, you are muted, but feel free to type questions into the questions for staff box throughout the presentation. At the end, I will read the questions for our presenter to answer. So with that, our presentation today is titled Global Marine Biodiversity Monitoring Through Partnership and Innovation, presented by Dr. Luke Thompson. So Luke, I'm going to go ahead and give you screen sharing permissions. And while you're getting your presentation set up, I will introduce you to our audience. Okay, so Dr. Luke Thompson is an associate research professor at the Northern Gulf Institute at Mississippi State University and NOAA's Atlantic Oceanographic and Meteorological Laboratory, or AOML, in Miami. Luke received his bachelor's degree from Stanford University and his PhD from MIT, both in biology. His current research focuses on marine systems from microbes to fish to mammals, using omics methods, especially DNA sequencing. His lab is developing methods to facilitate high-throughput environmental DNA sample processing and data analysis, and applying them to monitoring and conservation efforts in the Atlantic Ocean, Gulf of Mexico, and the Great Lakes. So Luke, thank you so much for presenting today. I will now pass it off to you, and we can see your slides as well.
Great, thank you, Catherine. Thanks for the invitation and the kind introduction. So yeah, today I want to talk about some of our work at AOML, and in particular focused on global marine biodiversity monitoring and the partnerships and innovations that make that happen. Please disregard the little header there at the top. I neglected to update that. So today is July 20th, if you're keeping track at home. And yeah, as Catherine said, I'm at NGI, which is a NOAA Cooperative Institute through Mississippi State University, and I'm based at the Atlantic Oceanographic and Meteorological Laboratory in Miami. So to orient you a little bit, this is what our lab looks like. It's on Virginia Key off the coast of downtown Miami. It's this white building there circled in pink. and it's a great location. We're right on the Atlantic Ocean and Biscayne Bay. We're located across the street from the University of Miami, which has a lot of additional resources for marine science. And yeah, it's a great place to work. In fact, we're actually in the process of hiring a couple new postdocs and a technician and a project manager, if we can get that all through the system. So if you're interested in the work I'm talking about and you want to get involved, please let me know. And we have some additional projects related to carbon export and ocean acidification that I won't really even touch on today. So there's a lot going on. So as a background to my talk, I wanted to just lay out a few things. I'll go through this visually in the next few minutes. So the main thing sort of in terms of background is that we need high quality assessments of biodiversity to understand marine biogeochemistry and to conserve communities. We need to know what's there to be able to obviously to understand it and to protect it and conserve it and be able to measure it over time. And so DNA sequencing has emerged as a key tool for monitoring biodiversity. And it's also being coupled with high throughput imaging and remote sensing. And that way we can get a really comprehensive profile of marine communities. And there are some efforts underway to do this, but there's a lot of room for improvement and expansion. So a couple of the networks that we're involved with that I'll talk about today are MBON, which is the Marine Biodiversity Observation Network, and that's partnering with the UN Ocean Decade and Marine Life 2030, and that is really expanding our DNA and remote sensing based observing. The second program that we're involved with is called biogeochemistry, and hopefully a lot of you have heard of GOMECC, which is the Global Ocean Ship-based Hydrographic Investigations Program, and this is adding both the biogeochemistry part of GOMECC is adding both DNA and imaging-based biological sampling to this long-standing global ocean monitoring program, and I'll talk about both MBON and biogeochemistry today. So a little bit about Marine Life 2030, and I want to thank Enrique Montes at AOML for providing some of these slides. So the problem or the situation is that at present there is no systematic effort coordinated internationally to observe life in the sea. And so the goal of this effort is, or the goal of this this effort is to enable efforts to characterize how marine biodiversity is changing, how it affects us, and how to guide sustainable development. And so as a key example of this and where we're lacking, this is a beautiful picture of the Earth and the ocean showing chlorophyll or ocean color for the ocean and vegetation for land. And so this shows you where there's the most primary productivity and that sort of thing. And that's, of course, really important information. But that's really the only inputs into a lot of our models for how we understand the ocean in a larger context when we integrate with fields outside of biology. And so this is definitely something that we want to improve upon. And so, for example, we do have some biodiversity data that's stored. There's a very nice system called the Ocean Biodiversity Information System, and it's containing a lot of records about observations of different species and so forth in the ocean. And but it's not complete. As you can see in the map, there's some parts that are pretty densely sampled. There are other parts of the ocean that are very minimally or not sampled at all. There's been a drop-off recently in the number of records over time, as well as at certain latitudes there are fewer records. So this is certainly something where we want to improve coverage, and the baseline that is the total coverage at a given time point, that to then track over time changes in diversity, abundance, ranges, and connectivity between communities. Another example from OBIS is that as you get deeper in the water column, there's fewer observations. So most of the observations are confined to the surface. So to kind of go through the challenges then in this situation for global ocean biodiversity sampling, there's inconsistent sampling programs, which I kind of showed you a little bit already. There's also a lack of standardized sampling and DNA extraction methods. So when we're specifically talking about omics-based methods or genomic-based methods, that comes into play. Also thinking about genomics specifically, there's incomplete reference databases, and this is true actually for both genetic and optical data, which I'll talk about a little bit in a little bit. There's also a lack of analytical reproducibility, people using different methods, and also not even just reproducibility necessarily, but also just sensitivity and accuracy of the methods that are analyzing the sequence data and the optical data. And then there's sort of underdeveloped machine learning and modeling methods. There's a lot of capability now in coming from other fields in natural language processing and image processing and basic research and machine learning and weather modeling. There's a lot of other areas where these methods are used more than they are in biodiversity and genomics and marine imaging and kind of our neck of the woods. So there's a lot of potential there that's untapped. So first I'll talk about standardized sampling and extraction methods that we've been developing in our lab. So these are some results from our lab that we'll talk about. And so in this case, the goal or the idea is to use a standard filtration method and one that's emerged that's very commonly used is called Sterivex. I wouldn't necessarily say that we should use this for all ocean sampling because there are reasons to use other types of filters, but this is one that's emerged as a popular option, and so that's the one we're using here. And we want to have a high throughput and reproducible method. So the Sterivex are these little filter cartridges shown in the center. These can be, you can filter water through them, and I don't have a picture of the pump that we use, but we use a peristaltic pump to filter water from either an Iskin bottle that's dropped on a line or on a CTD rosette shown on the left, or collected via an automatic sampler like the SASE that's shown on the lower right, and I'll talk about that a little bit later. And so however you get the water, you then filter it through one of these Sterivex filters, and you maybe add preservative or maybe just run it dry, which is our preference if we can. But now you have this problem of how to get the DNA off of the filter. So the traditional method was to cut those open and to peel off the filter and cut it into pieces and then stick it into an Eppendorf tube. And it's very tedious, it's prone to contamination, and it takes a long time. So there's been a newer method that was developed where you add like glass beads basically to the inside of the filter. You can do this either before or after you collect your sample. We do it after, and then you do a vortexing step, and then that will kind of chop up the cells and give you a nice lysate. So we found. So what we did in our group, and this work was led by Sean Anderson, is to test this for some samples that were collected near AOML. So we used two different we used two different platforms. This is showing one platform, two images of it. So on the left, you can see the Kingfisher Flex robot without any plates on it, but this is a magnetic bead handling robot that we use to increase our throughput. And on the right, you can see inside when it's loaded with samples, it uses magnetic beads that will bind to the DNA in the the lysate from the sample, and then do a series of extraction and washing steps. And what you're left with at the end is purified DNA attached to beads that you can then remove the magnetic beads and just have your purified DNA. So it's a really nice platform. And so what Sean did was he tested with or without beads or a different combination of beads, and then we looked at the 16S rRNA gene of bacteria and the 12S rRNA gene of fish. So we'll show you the fish genes today, the fish results. The high-level results was that generally all treatments gave us similar concentrations of DNA, but the treatments were had slightly different taxonomic composition, and we got better recovery of hard to lyse organisms like diatoms with beads. So using both kinds of beads in particular was the best, and so we recommend using both large beads or large and small beads together to get the highest recovery of the broadest diversity. And so an interesting thing that happened with this survey is that our samples were taken from the Rasmus dock, which is shown on the right there, and it's close to the Miami Seaquarium on the bottom, which will become relevant in a minute. Because when we looked at the sequences, we had a really interesting pattern where we saw some fish that weren't expected, such as rainbow trout, herring, and smelt. And make a long story short, we surveyed community of local scientists and discovered that these fish were being thrown out from the seaquarium, and so these were fed to the marine mammals, and then the leftovers were dumped in the water, and that current was going from the bottom of the screen to the right side of the screen, and we were picking up this environmental DNA from these fish. So it was a nice confirmation of the method, because even though we didn't expect to see these species, we could determine that they were being put into the water directly. And so so this is a good confirmation of our fish sequencing method, as well as the extraction method, which we can use in open ocean context as well as in coastal environments. And we can use it both for collecting and analyzing fish DNA, as well as microbes and plankton. So another challenge that I mentioned is reference databases, and there's two challenges here. One is that we're missing taxa from some of these reference databases, and so we're tackling that by sequencing taxa that are missing. And so we basically, if we know there's something that is in the water that isn't in the database, that's obviously a red flag, and we want to sequence those. But if we also see a sequence that is highly abundant and it doesn't match something exactly, we might be able to determine where it might have come from based on similarity, and then go ahead and target those. So I don't have a particular slide on sequencing missing taxa, but this is something that we're working on. And the second aspect is improving the usability of reference databases that are already out there. And so this is, I'll talk about our efforts on a fish-specific database, but these kind of tools are relevant to all the different taxonomic groups, whether it's plankton or bacteria or fish. So this was led by Jean Lim in my group, and she developed this tool called MitoHelper. So what this does is it uses the popular mitofish database from Japan, which is a nice database, but needs some curation. It doesn't necessarily link well to the US NCBI database. Sometimes it's unclear whether you have a full-length reference sequence or just a part of it, or maybe the whole mitogenome, or anything in between. It also, you sometimes want to get more information about the sequences than you can get. So there's a couple of different tools or commands that MitoHelper provides to pull down records as well as alignments of these reference sequences. And so this has been, I think, a useful tool for folks, and again, provides, it's useful for fish sequences in particular, but I think this framework would be relevant to other reference databases as well. Another area where we're working to improve kind of the usability of genetic data from the ocean is developing workflows. And so this is a way to take the kind of commands and work that you would normally do with a genetic analysis of a marine environment, and to make those commands more rapid to deploy, more iterable, which is to say to link or to provide reproducible outputs. Every time you run the workflow, you get the same outputs that are in the same folders. You can open them the same way, and you can write programs that will take the output and do something with it. So everything is named for you. Everything is done automatically. You can go back and change things if you want. But so we used something called SnakeMake, which is a workflow. And what that does is basically you provide it with a configuration file, a list of your sequences, or a list of your sequence files, and a reference database. And then it will do the rest. It will run these diversity and taxonomy and commands, and then generate a report. So it's a really handy tool. And this is now coming out in GigaScience this year. So that should be 2022, but should be out in the next week or two. Another nice feature of this tourmaline workflow is that it can be run using a Docker container. And so Docker is you can think of it like a virtual machine, or it's a system that you can you download the software, but then you can run the container and then download the container from a server, and then you can run that container locally on any computer, whether it's a Linux PC or a Mac. And the idea is that you get the same experience no matter what system you're working on, because you're working inside this virtual machine. So Tourmaline comes with this container that it runs inside, and so anyone can run it. You don't need to install the software on your own computer, although you can. It works both ways. Another sort of workflow that we're developing, it's currently more of a tutorial, but this is a metagenomics tutorial. So I don't think I said, but Tourmaline is as an amplicon sequencing workflow, or a metabarcoding. So metabarcoding is where we sequence a single gene from all the organisms in a sample, and a metagenomics is where we sequence all the DNA from everything. So it's a more complicated analysis. But Nastasia Patton and our group at AOML have been developing a metagenomics tutorial that walks the user through an analysis. And Nastasia gave a great presentation a few months ago, and there's a video recorded of it. So she really provided the background for how you do this, and then we've put the commands and instructions in a single markdown document. And we're working on this now, and the idea is that it'll be both a tutorial for people to learn metagenomics, but also potentially we could turn this into a workflow as well, so you can actually run it on your samples. And then another technical thing that we're developing are algorithms to improve taxonomic assignment of sequences, as well as to model biodiversity with environmental parameters. And eventually we want to create composite indices from multiple measures of biodiversity. So getting started, we've been working on some machine learning algorithms for taxonomic classification from metagenomes. So this kind of would fit into that that tutorial I just mentioned on metagenomics, one part of analysis of metagenomes is to figure out what are the taxonomic groups in that sample. And so what we've done, and this has been led by Jean Lim in my group, is to well, when you do sequencing of microbes in particular, you can think of each microbe having a genome represented by a different colored circle. So you chop up the DNA and then you align them and or otherwise classify them, try to figure out which microbe they came from by comparing them to reference databases in some way or another. But there's lots of different ways to do that. So and I've included this figure on the right, which shows that when you have a sequence of DNA, you typically, or a piece of DNA, you typically sequence the two ends of it, and then you have some part in the middle that's unsequenced. So this is called paired end sequencing. And so when we are trying to classify our sequences, we have to deal with that. So what we've done is we've been using this algorithm called Deep Microbes. And this is a taxonomic classification algorithm that uses some deep learning methods such as long short-term memory and including deep neural network training, and also uses a k-mer-based approach, which is where you break up the sequence into lots of different pieces. So Jean and an intern working with us, Helen Park, have been testing this out, training this algorithm, or training this model using marine metagenomes, because this program was developed for gut metagenomes. So Helen and Jean are using this with marine metagenomes, and then training the model and then testing it on different data sets. Meanwhile, we've got a group of researchers that was organized around an NVIDIA hackathon back in February. And this group has continued to meet, and we've got several different algorithms that the group came up with using various machine learning algorithms, one based on images, another based on k-mers, which are those short sequences of letters, another based on the BERT transformation, which is what like Google uses to suggest like what's the next word you're going to type, that sort of thing. And then we're comparing that to a standard database, like using a full database and k-mer-based approach, a method called Kraken-2. And so we have some different algorithms that we're comparing. And so the end goal of this is hopefully that we'll have multiple methods for analyzing or determining the taxonomic composition of a microbial samples from the ocean, and potentially a better algorithm in some cases that might improve on existing technologies. And so now I wanted to turn to some of those sampling efforts where we're going to start applying. Eventually, when we get sequence data, we'll start applying those methods to these surveys, and that are going to give us a wider view of the ocean. And I'll talk a little bit about what kind of information we can get from those surveys, and then I'll conclude with some results closer to home from the Gulf of Mexico, where we're a little bit further along in doing some of this work. So first of all, MBON, the Marine Biodiversity Observation Network, the goal of MBON, or the vision, is to develop a community practice to understand changes in marine biodiversity. So the idea is ocean observing systems and networks of different groups and scientists around the globe, both coastal, EEZ, and the high seas. So everything is fair game. There's a focus on marine protected areas, and then focus on community resilience in the blue economy. So there's an economic component to this as well. And so MBON has been around for a while, and it's continued to grow over the last few decades. to the point now where there's a lot of buy-in from the United Nations, from different organizations. One that I wanted to mention was OBON, which is the Omics Biodiversity Observation Network. We also have GOOS, IUS, involvement from BOEM, GeoBon, NASA, and so it's definitely a widely there's a lot of buy-in, a lot of participation across these different communities, and so and especially with the UN decade happening now, it's an exciting time to really spark these collaborations. So one aspect of MBON is the use of these GOOS biology and ecosystem essential ocean variables, or EOVs. So there's a variety of different functional groups or different taxonomic groups, as well as different habitats that are focused upon. And so the way that this observing network is meant to work is where we kind of have a feedback where we have requirements for how observations are made and reported. Those observations are made, which leads to data products and sharing across different communities and end users and stakeholders, and that then builds this coherent, consistent, and coordinated ocean observing system, which then feeds back and improves requirements and improves the next round of observations. So put another way, from the data and data sets that we collect, and we can slice those or filter them by taxonomy or space or time, we get maps of abundance, and as well we can graph trends and see how things are changing over time. And so an example of these of MBON would be the South Florida MBON. So this is the South Florida MBON stations are shown in the map in orange, but they're very close to the NOAA iconic reef sites, where there are some reef restoration, as well as a couple other sampling efforts shown on the map. And so one of the things we're starting to do is sample at these sites, in particular the iconic reef sites, over time to determine how the reef environment is changing and how the biodiversity is changing as we restore reefs. And so one of the things we're doing is using eDNA as well as satellite remote sensing to monitor those changes. And so we've started this project now. We got some seed funding to do eDNA sampling on reefs, and so we're using the subsurface automated sampler for eDNA, or SASE, that was developed by Nate Fromell and Ian Enochs at AOML. And so this is a little peristaltic pump that holds one of those Steravex filters I talked about earlier. So actually this afternoon we're doing one of our first pilot experiments in the field, and our hope is that we can get these working. They work fine now, but we haven't done a lot of testing in the environment, so get them environment tested, get some data back, and then by next year we'll be ready to do some sampling on the iconic reef sites and start to monitor how the community of fish and invertebrates is changing with restoration. And then I want to talk about biogeochemistry. So before I talk about biogeochemistry specifically, I want to talk about a related effort that I think is really important to thinking about what's possible with biogeochemistry. So this is related to the Tara Oceans expedition. So Tara Oceans is a French and European-led effort to to monitor biodiversity around the ocean using a lot of technology, a lot of different imaging as well as genomics methods. And so this is a figure from a paper published by some of the Tara people in Frontiers, and it's about coupling imaging and omics in plankton surveys. So from these different Tara sites, they have a ton of data, various different imaging methods, and that target different size fractions of plankton, as well as the omics part of it. So so they've really done some cool stuff with this data. So one of the things they developed is this eco-taxa program that allows them to annotate the imagery data, and then they can validate that with genetic data. So there's a lot of technology that goes into this, and so they've used this to do some cool stuff. So they had a paper out last year, I believe, in Nature Communications, where they analyzed just a ton of data from all these different stations. And so on the lower left is showing you using the omics method the different taxa that they pulled out, and the ones that are colored they could identify. So there's some different, in particular, this is focused on diazotrophs or nitrogen fixing plankton. And then they compared on the right, they compared the relative abundance from the genomic methods versus the relative abundance with the imaging methods, and you know, they're not perfectly aligned, but there's some good overlap, and so they're showing that, you know, these two methods are both you know, effective and complementary to getting a fuller picture of plankton in the ocean. Switching gears a little bit, another thing you can do with marine omics or marine genomics is you can detect the relationship between nutrient levels and the genetic functions that are involved in those nutrients. So this is a paper that I wrote several years ago from some data in the Red Sea. So on the right is a kind of a contour map down the water column of the Red Sea from the surface to 500 meters, and each of those circles or concentric circles is a sample where we took environmental metadata like nutrients as well as metagenomes, and then the colors show on the outer circle the phosphate concentration, and on the inner circle the sort of relative abundance of the phosphate transporter, the gene for the phosphate transporter PSTS. So what it shows is that there's an inverse relationship between the phosphate concentration and the likelihood of microbes having the gene for transporting phosphate. So when phosphate is low, microbes are more likely to have this transporter because they need it to transport phosphate is the hypothesis. Whereas when phosphate is high, the need for this like high affinity transporter is not as much. And the same information is shown on the right, just in a graph form. And so the very clear relationship between phosphate and this gene. So this is a single example, but then Adam Martini's group at UC Irvine, and Adam is a collaborator on the biogeochemistry project. They've sort of expanded this idea to look at, in this case, it's looking at Prochlorococcus across the global ocean. So on the graph here, you can see latitude in the Pacific and then in the Atlantic, and there's these really striking trends of phosphate concentrations and then ecotypes or kind of groups of Prochlorococcus that correlate really well with phosphate as well as temperature. And so there's a lot you can do with these ocean transects, like we want to do like we're doing in biogeochemistry. And so what Adam's group also did was they took some GOMECC think they did one or two, yeah, a couple of GOMECC lines, maybe three, P18 and I07 and 09. They did those bioGOMECC lines, and they also got samples from biogeotraces as well as Tara that I just talked about. And they sequenced metagenomes from those. And so one of the things that you can see in this graph on the right is that the resolution of bioGOMECC is much greater because of the way the sampling is done, and we can sample continuously, kind of, every few hours as the ship is going. We can just get water from the ship's flow-through system. We can get a lot higher resolution in bioGOMECC than we can in some of these other operations. Maybe we don't get quite the abundance of different kinds of data, but we can get a lot better resolution, which provides opportunity for some cool analysis. So Adam's group then took that data, and they had this really nice paper in Science where they were able to determine the nutrient limitation regimes of different parts of the ocean. So these lines shown in the map on the right are the same lines that I just showed you, but now they're colored by whether the microbes, and this based on these genetic measures like I showed from the Red Sea, which samples are nitrogen limited or co-limited, nitrogen and iron, or nitrogen and phosphorus, or phosphorus limited, or iron limited, or iron and phosphorus. So this is the kind of thing that's only possible with global sampling of the scale, like GOMECC. And then just another way to look at it is, you can look at the map and based on what we know about parts of the ocean that weren't sampled, we can extrapolate, which parts of the ocean are under iron stress, phosphorus stress, or nitrogen stress. And then, of course, there's some co-limitation in certain parts where those regions overlap. So with bioGOMECC, we want to expand this. So my map got covered a little bit, but there's around 66 lines in the GOMECC program. With the bioGOMECC pilot, we've got 3 or 4 lines that we're going to do. And in addition to metagenomic sequencing, like Adam did with those other cruises, we're going to add a variety of imaging methods similar to what the Tara group is doing, and that allows us to combine these different methods in really new ways to look at relationships between these genomic measures as well with things like particle size, optical properties, phytoplankton abundances and diversity, and then come up with, based on all that data, come up with models and indices of diversity, and how come up with ways of representing that diversity in using multiple metrics. That'll be more powerful, more robust, and more informative for things like conservation and modeling. And so finally, I wanted to talk a little bit about some of our work closer to home, looking at the Gulf of Mexico and what we can say about some of the patterns we're seeing here. So this one is from the Southeast Area Monitoring and Assessment Program, or SEAMAP. So this is a survey that's been going on for 30 years, and they're using camera traps, like shown in the picture there. So when at different intervals, these bottles are closed while the camera trap is down on the seafloor. So it's taking video of fish that swim by, and then we can close those bottles, and we can measure both fish eDNA as well as plankton and microbial DNA to get a picture of both, how well the fish compare to what's in those bottles, as well as how that relates to the lower trophic levels. So we are still getting the last of our data sequenced from 2021. So what I'll talk about today is is the data that Sean has analyzed from 2019. We've got sites from Florida and the Texas shelf, surface and bottom using the CTD. And these were all sampled using that Stereovex filtration method like I talked about earlier, that's shown in the lower right. So we've got, yeah, again, stations from Key West, Tampa area, Panama City, and Florida shelf, Galveston, as well as Brownsville, so Texas shelf. And so we see a really clear pattern between surface and bottom, as we expect. There's lower diversity in the surface than in the bottom depths, and they're clearly differentiated from each other. And we can determine that oxygen and temperature are driving a lot of the patterns and separation. So at the surface, we see pretty clear delineation between the different regions, and so that's suggestive of some regional separation among these different parts of the Gulf. But in the fish data, our first look at the fish data, and we're getting more this month. The initial look was that there wasn't a dramatic structuring, but we're still digging into the data and comparing that to the camera data. So stay tuned for updates on that. And then finally, we have some data that we've been working on that dates back to the Deepwater Horizon spill. And so Gene Lim has been working on this with me. And so we have samples from around the wellhead area, and this were taken during or after the spill, as well as samples that were enriched with oil and to try to model the spill. And the whole idea of this project, of course, the Deepwater Horizon spill was a horrible disaster, but we wanted to try to figure out what was the dominant microbes that were degrading the oil, and could we detect them before the oil. And essentially, we could not find the exact microbes before the spill that are now thought to be the ones that were degrading the oil after the spill. There's just very little abundance, and that's interesting because it suggests that, I mean, we know they had to be there, right? They started growing when the oil showed up, but they were at such low abundance that they couldn't be detected before the spill. And this kind of gets at this idea in microbial ecology. It's called the Bos-Becking hypothesis, that everything is everywhere but the environment selects. And so those microbes were present, but they couldn't be detected because they were in such low abundance. They hadn't been selected by the environment. So in any case, what I'll just show here is that Gene's been using some different tools to study these metagenomes and seeing some interesting patterns. So this one is based on a tool called SingleM that computes taxonomic abundances from reads based on single copy marker genes. This is based on a gene called RPLB, and then we can do an ordination on these, and so we see some interesting patterns, but importantly, the pre-spill samples, they look like any other surface or epipelagic or mesopelagic metagenome from the Gulf of Mexico. These Tara ones in green are also from the Gulf of Mexico from a little bit later after the spill, but still, normal looking metagenomes. And so from both this analysis and a more in-depth analysis using read mapping, there was no pattern of sort of being able to predict those functions of degrading oil before the spill happened. We know they had to be there because they started growing and were able to degrade the oil relatively well once the oil showed up, but in a normal seawater environment in the Gulf of Mexico, unless you're closer to hydrocarbon seeps and that sort of thing, they're pretty low abundance. And so that's all I have for today. I wanted to thank a lot of different folks at NOAA and collaborators for their help and contributing to the science that I shared today. And especially want to thank a number of different NOAA line offices and programs that have contributed to this work. In particular OAR, NGI, and I should say OAR, NOER, as well as NGI, GOMECC, Ocean Acidification Program, and then our collaborators at MBARI. So yeah, I will, I'll leave my slides up, I guess, in case there's questions, but I'm happy to answer questions in the chat or out loud.
Excellent. Thank you so much, Luke. That was a fantastic presentation. Yeah, folks, feel free to type questions into the questions for staff box, and I'll read them out loud for Luke to answer. We do have one question that came in earlier, Luke. This one's from Carol. She says, Luke, great talk. Please tell again what your differentiation is between metagenomics and multi-gene, multi-sample metabarcoding.
Yeah. So yeah, with metabarcoding, you can do multiple genes or multiple targets. So imagine you have a seawater sample and you've extracted all the DNA from it, and so it should be full of bacteria and plankton and maybe some traces of fish and whales and viruses, and anything that might have ever been in that water could be in that DNA sample. So with metabarcoding, you can choose which gene you want to target. Say you want to go after bacteria, so you use the 16S rRNA, the small subunit, or you want to go after fish, so you choose the 12S rRNA, which is also the small subunit, but of mitochondria of fish, or etc. So each one of those will give you a different picture, because those primers will ideally only amplify that taxonomic group. So you can kind of, I mean, you can analyze those data sets together. You can look at co-occurrence patterns. There's a little bit of, apples and oranges, but they are each a profile of a given taxonomic group, or large taxonomic group. And then with metagenomics, you've kind of got everything there. So you might miss out on some of the rarer stuff, like fish, for example. Those might not show up just because you have so much more bacterial DNA. But then, okay, maybe you do have everything there at some level, and then you're still going to need to do some sort of profiling. So that will also be dependent on the database, and if you do assembly, you're probably more likely to assemble those bacteria or things that are more abundant. So I don't know if I answered the question, but basically they're all getting at some of the same things, but you can do more with metagenomics, but you're gonna potentially miss out on the rarer stuff, and you're gonna need to do a lot more. The algorithms are more, there's many more algorithms, and they're more complicated, but ultimately it can be more rewarding because you can get functional information as well as taxonomic information, and that's pretty useful.
Excellent. Thanks so much, Luke. We had another question that came in. This one's from Victoria. She says, hello, Luke. Great talk. Thank you for your time. How can we get in contact regarding your work and joining your team?
Oh, great. Yeah, just send me an email. I can put it in the chat. Would I do this to organizers and panelists, or Catherine?
Yep, send it out to all attendees.
Okay, I'm not seeing that as an option. I see organizers and panelists.
Oh, I can put your email here in the chat, Luke. So
Okay.
I got it. Do we have other questions from folks in the audience? Okay, I'm not seeing any come in, but here's Luke's email. Oh, okay, I see we both put it in at the same time. Okay, good.
Oh, great. Yeah, so as I mentioned at the beginning, we're hiring. So we've got one postdoc that's going to be focused on the biogeochemistry and MBON work. And then we have another project on sediment traps to study the biological carbon pump. And we have there's some links there to ocean acidification, foraminifera, some really cool angles there. That's a joint project with Emily Osborne at AOML. And so we have a postdoc to work on that project as well. In addition, we're going to be hiring a project manager who's more of a bioinformatics PhD level, but sort of floating between different projects and running our HPC effort using some of the supercomputer we have at Mississippi State, Orion. So that'll be a third position that's more of a project manager, but heavily bioinformatics. And yeah, we're also looking to hire a technician who's going to be doing some of the SASE work and general molecular biology stuff in the lab as well.
Awesome, thanks, Luke. I actually just put a link to AOML's career page in the chat if folks want to flag that. I think a lot of the positions supporting or working at AOML will pop up on this page. And I think folks can also check USA Jobs and the University of Miami career websites to look at these upcoming positions.
And also Mississippi State job board, because those first three positions will be through Mississippi State, but they'd be based in Miami. So just like me, I'm a Mississippi State employee, but I'm based in Miami.
Awesome. Okay. I don't see any other questions here, but if folks do have lingering questions, please reach directly out to Luke. His email is here in the chat. And so Luke, just want to say thank you so much again for presenting today for the seminar series. And I want to thank everybody for joining us today. Our next seminar is on August 19th at 1 p.m. Eastern time. So this is different from our normal seminar time, which we have it normally is at 12 p.m. So it's at 1 p.m. August 19th. Please visit the Omics website to get information on how to register for that talk. This one is going to be by Dr. Ingrid Spies from NOAA's Alaska Fisheries Science Center. So thank you all again for joining today. Have a great rest of your day.
Thank you, Catherine. Thanks, everyone.